Okay, thank you. Thanks, Vasily. So yeah, I have kind of like a varied background. So I started as a mechanical engineer and then finally ended up being a biomedical engineer. Now, so I got trained in an institution where we used to do more translational research. So the way I approach research is not from more the fundamental understanding, but more from the need. So today I'm going to discuss that approach and then also show you some results that we got over the years. So this kind of started in 2012, 2013, and it's like 10 years of work that I'm going to discuss today. So again, so this is mainly focused on portable brain imaging and portable brain imaging application in acute brain injuries. Um, Okay, let's see if I can. Okay, so now this need-based design process. So this is, I think, mostly popularized by this Stanford Biodesign program, where the idea is to kind of first identify a clinical need. So we mostly know that there is some need, and that's essentially kind of gives us kind of a broad picture. However, specifically what's needed in terms of technology development that needs so that has to go through a certain process where we talk with clinicians, we talk with the stakeholders, the patients, also maybe general population, and then that provides us a bunch of different need, like technological need, that we can then satisfy with our engineering background. So that's where the concept generation comes and the concept selection comes. So I'm today primarily going to focus on this first two part, which is the identification of the need. So this is again a need in acute brain injury, and to see, and the need again is on portable brain imaging, but specifically what? So that's what I'm going to discuss first, and then to kind of show you how or what we invented to satisfy that need, and then to give some final results on the prototype and also the biomarker that is currently going through a clinical trial. So okay, so that's kind of the background of the design process. Okay, so now, so in 2006, there was this landmark experiment where so before this, actually, there was this clinical way of evaluating patients with acute brain injury, and then the idea was to classify them as responsive or not responsive. And those who were not responsive, they were considered or they were classified as vegetative state. So now, so there was always this debate going on about whether this clinical evaluation was valid or to what extent it can identify the awareness in a patient. So in this particular study, this was again done in one person. So, and the idea was to image the brain with functional magnetic resonance imaging and to see if given a command, for example, ask the person to play tennis or imagine playing tennis, or ask the person to like walk around the house, like moving around home, and to see the brain activation in the fMRI scanner, and then to compare that brain activation with healthy controls or healthy volunteers. So interestingly, in this particular patient, they found that there was no difference. Even though the patient satisfied the criteria for clinical criteria for the diagnosis of vegetative state, there was no difference in the brain activation when the person was asked to play tennis or to move around the house. So this kind of brought up this idea that the clinicians need to be much more careful in evaluating this covert consciousness. Now, so this is 2006. So at that time, the functional MRI, fMRI was also kind of in it, is kind of was used in neurology. Mostly it was structural MRI that was used, and functional MRI was kind of catching up. So around that time, there was this idea that if these patients who are classified completely based on clinical diagnosis or clinical evaluation, and up to 15% of those patients show signs of covert consciousness, which is what is called cognitive motor dissociation, then we need to develop technologies to identify this awareness. Now, the challenge here is that a lot of these brain injuries in the acute stage, the consciousness is fluctuating. So if, for example, of course, at that time, fMRI was kind of established as a technology, was being increasingly being used in clinics. So the idea was like, so how often can we do fMRI and whether this is even feasible if to monitor someone continuously when the consciousness is kind of fluctuating after brain injury. And but then this kind of was driven more from the ethics side because this is kind of a decision that the clinician has to take to continue treatment or not. So this is a kind of a decision that needs all available resources to be used to make informed decision about the future of the patient when the patient is not showing clinically not showing any awareness or is not responding to commands. So now, so yeah, so this was when the experts kind of tried to also see how they should label or how they should classify. So there was this idea of whether to use vegetative state as the way to mention this particular brain state, or maybe unresponsive wakefulness syndrome is a better way because unresponsive wakefulness syndrome provides this possibility of cognitive motor dissociation, CMD. So this is where so my collaborator, he also kind of started thinking, and I was also at that time. Kind of I finished, I moved to Europe. At that time, I was also looking into 2010. I moved to Europe and I was also looking into a portable brain imaging, if that can satisfy the requirements. So now for the need finding, so this was like the initial stages of technology development and all, but before even the technology is applied to this particular problem, we have to also see what's kind of the public perception of this problem. So is it only a problem that clinicians think, or this is also in general public, there is a there is this need for technologies to identify or solve this kind of an issue in brain injuries. So so this was an online crowdsourcing study that we did. So we recruited 1,297 lay people from 32 countries, and then we investigated. Like in lay people, like for example, when you read a newspaper, you are given a case, a clinical case, which is kind of watered down a lot, and then you make a decision of whether, I mean, how you think about that case, right? So there are like a lot of these where you read in newspaper that someone with in a coma state, like so vegetative state, they responded to certain stimuli, they did an eye blink or they did something, moved a finger, and then I know like so there was this debate of whether we should continue because it's of course the public funds that's used to provide the life support to the person. So this was kind of the idea. So we wanted to see what the lay people think, general population think. So yeah, so the idea was to investigate using this online crowdsourcing platform on the attitude of the people on this particular topic. So we crowdsourced over like, so yes, 1,297 lay people from 32 countries. So the Europe, for example, United Kingdom, we had 560 participants overall from Europe, more than 1,000 participants. Also, there were participants from America, US 57, then Asia and Australia. And then you can see kind of like in the table, the distribution. So it's kind of equally male, female, age, about 30 years old, plus minus standard deviation 10 years, also comparable between male and female. The education level, you can see about half of them university educated or higher. Then we about 40% high school or college, and then less than high school is about 2%. So very small percentage of the total. Now, in terms of the employment, 40% were full-time employed, and then 47 half-time, and about 12% unemployed. Now, the religion aspect is important here. So how important is religion for these people who participate in this survey? So roughly half. So for half of the participants, religion was not important at all. And for other half, there was different levels of importance, and about 4% religion was extremely important. So you can see that, in terms of religion, also there was kind of a half-half distribution. And So what we found is very interesting. So first of all, so the way the study was done is the way you read a clinical case in newspaper. So we provided a clinical case, and then the participants, they had to kind of say whether they prefer the term vegetative state to define that case, or to classify that case, or they prefer unresponsive wakefulness syndrome to classify that case. And then, of course, you also have the option undecided. So what we found is about 60%, in fact, like the term unresponsive wakefulness syndrome to classify a coma state. And then the vegetative state label was about 24% wanted to use that term. So yeah, so that's interesting. So there was more lay people actually identified with unresponsive wakefulness syndrome, this particular term, to classify or to label coma. Then the second was to kind of then see in how um, so in those participants, how much possibility is there for this cognitive motor dissociation. So in these people who are in this coma state, how this lay people, they think about the possibility that they might be aware. So they might be aware, but not being able to not the clinical evaluation is not being able to detect the awareness. So it was interesting that those who preferred the term unresponsive wakefulness syndrome, they actually were more they see the more possibility of the person being in this aware covert conscious state than those who actually prefer the term vegetative state. So this actually kind of shows us that how we label things actually changes the perception and what's expected. So the kind of the behavior of a person. So this was an interesting. So we should be very careful in how we name things. So this was an idea that of course, it's in psychology is known, but we kind of saw that also in this particular case. Although the kind of the difference was small, so the coinc D is like 0.164, but there was a difference in this possibility of cognitive dissociation based on how they label that condition, so how they term that condition. So this was interesting. So this actually kind of showed that there is in general population this some awareness of this state, and of course the technologies can then provide objective measure. So that was then the idea of the project. Now, so this kind of motivated us to develop an objective method that can be used at bedside to provide feedback of consciousness. Now, why this is important is that so after brain injury, so when these individuals are in this kind of an unresponsive wakefulness syndrome kind of a state, then for recovery of consciousness, a lot of the stimulation is done at the bedside. So it's usually by the caregiver or the family, the touch, hearing, vision, smell, and all. But the challenge is that, so in fact, I have seen that in ICUs, that there is no feedback that's given. So for example, there is ECG, so the heart feedback is there, and they can see that on the screen. The PPG is there, pulse oximetry is there, but how the brain is responding to the stimulation, there is no feedback. So we used to kind of at that time only kind of realize that if we kind of give some feedback as the caregiver or the family, wife, husband are interacting with the patient, if we can provide some feedback of the brain, then that would be very useful to motivate them, in fact, to interact with the patient more, because a lot of time there's pretty much no like facial expression change or anything, because these individuals are completely unresponsive. But they do have reflexes, so sometimes the reflexes kick in, but no kind of a explicit response to the stimuli. So that was also kind of a motivation for us to develop this brain imaging, kind of to provide it at the bedside to give feedback to the family and the caregiver. So yeah, so that's the idea from the neurofeedback side, but also this is a tool that we wanted to develop for the clinicians to make an informed decision. So the clinical technique needs a motor function. So the person needs to move a arm or a finger or to show a response. But many of them may have cognitive consciousness, so they may understand the command but may not have the motor action or the ability to produce motor action. So that's a challenge that we can solve with brain imaging. And then again, like I said earlier, so 15% of people who are in this kind of clinically classified vegetative state, they can perform mental imagery tasks. And this again may be detected with brain imaging. So this was kind of the motivation for us um for this need. Now, so this is kind of the initial work of need finding. It kind of took few years. Then after that, so we then try to identify what are the disease fundamentals that would give us a technique that we can then use for developing this portable brain imaging. So now again, the landmark study in 2006 was using functional MRI, and of course, functional MRI is depends on the change in the blood oxygenation, and that is related to the neural activity. So it's not a direct measure of neural activity, but there is a relation to neural activity. So that's what's called neurovascular coupling. So we then found, okay, so if fMRI is using this kind of this neurovascular coupling phenomena to detect the brain state, so what if we have a method to capture this neurovascular coupling? And this actually also came from the acute brain injury literature that actually showed that in the acute and subacute stage, there is actually a deficit in neurovascular coupling. So this is usually done with fMRI with EEG, combined fMRI with EEG. And then many times you actually may have a neural activity that you detect with EEG, but neurovascular coupling has a deficit, and then you don't see kind of a large change or a good SNR in the hemodynamic response part. So it's not only uh, the how the neurons are responding, but also how the neurovascular coupling is. That actually determines how well the fMRI signal is. And especially when you are running stats, the SNR becomes very important. So yeah, so that's why we planned on combining this NIRS, which is, I will going to discuss this next slide, but this is an optical measuring technique which measures the hemodynamics, the oxyhemoglobin, and then the EEG, which measures the neural activity. And then if we have this kind of a multimodal measure, then we kind of capture the complete system, not just the hemodynamics or not just the neural activity. And then there was this hypothesis, this is more for research from the clinician side, is to kind of see if this neurovascular coupling is a biomarker and the recovery of consciousness is kind of somehow linked to the recovery of neurovascular coupling. So this was the kind of like more scientific hypothesis to be tested with this study. Now, so like I said, so there was this then idea of from the disease state fundamentals to use a multimodal approach to get the complete picture of the tissue state or the brain state. So like I said, so functional infrared spectroscopy is was already a mature technique. So we don't want to go into kind of, uh, building something from scratch, because we are doing a translational research. So at that time, we identified FNIRS to be mature enough. It's 2013, 14 is when we started kind of writing the IRB and all. So at that time, we found FNIRS to be already being matured enough. And what it does is it's a very simple technique. So it essentially shines a light, which is a near infrared in the near infrared spectrum. It goes through the tissue, gets scattered, it's scattered back, and then you have a detector that detects the light, and then some of the light gets absorbed by the tissue. And this is this absorption is different between oxy and deoxyhemoglobin, difference in terms of which wavelengths are absorbed. So you can then, using this scattered light, you can estimate the change in oxy-deoxyhemoglobin in the tissue. So that's the functional near infrared spectroscopy. And then, of course, EEG was pretty well established at that time. It's already was used in clinics, and it's essentially antenna. So it's a metal antenna that you put on the scalp, and it detects this electric field changes that are in the neurons as they fire. So the neural firing creates a transmembrane current. The current flow, as we know, creates a field, electric field, and that's what is measured by this metal discs or antennas. So now these two techniques can be combined very easily, and you can combine it with a cap. And if you can measure them simultaneously, then you can identify the coupling between the two. So how they are interacting with each other, which is what, like I mentioned earlier, gives an estimate of the neurovascular coupling. So that was the kind of idea to go for this kind of a multimodal brain imaging. Now, so at that time, I was 2016. I joined University of Buffalo. So at that time, the study was then launched at the University of Buffalo. So yeah, so this was kind of like the feasibility study initially. Like I said earlier, so there was this clinical challenge that neurological assessments often misclassifies unresponsive patients as being in vegetative state. And then there was this logistical challenge with fMRI because it's like an expensive technique and it's difficult for a patient with all this life support to be moved to an fMRI scanner in an ICU setting. We tested the feasibility of this portable brain imaging, which is the FNIRS and EEG, if it's even feasible to be used in this patients in ICU. So the primary objective, of course, was to assess these patients for preserved consciousness. And then here, this was not a randomized control because this is the kind of like the feasibility study. So we did like a serial multimodal evaluations. It's a convenience sampling. So as the patients come, you put the scanner or the cap, and then you record data. So you record for active paradigm, passive paradigm. So by active paradigm, I mean when the patient is asked to actively move their limb. Passive is when they do mental imagery, and then also resting state paradigm. So we did. So this was kind of a long study that we started in 2016 after I joined University of Buffalo. And what we did at University of Buffalo is to bench test the system on healthy controls, because before we can apply it on patients, and by patients, I mean we had awake, responsive patients who were controls, but also unresponsive ICU patients who were in this kind of a coma state or in disorders of consciousness. So before we apply to those patients, we also wanted to see initially if it's on healthy control, how well it works. By that I mean, like, when someone does a motor action, how well can we detect the brain activity? When someone does a motor imagery, like a finger tapping, how well we can detect the brain activity. So this was done at University of Buffalo. And then, of course, from the scientific side, the question was the mechanism of the recovery of consciousness, and this is where there was this hypothesis that neurovascular coupling may be like a biomarker. So yes, this is how the study started. Now, so to start with, so I'm going to only so this is a published result, so I'm going to show only the resting state data. We are currently have few papers under review for the task paradigms. So this is where we did so a completely resting state assessment with this multimodal brain imaging technique. This was done at the bedside, and this was a convenience sample. So as the patients came, we imaged them in a neuro ICU or a cardiac ICU after brain injury. So this was done in Denmark, 9 subjects. So the exclusion criteria was that they shouldn't have any scalp injury or any acute life-threatening cardiovascular instability. And then also we had conscious patients. So these are patients who have recovered consciousness or who actually are in this kind of a neurological step-down unit. And they served as control. So we had 14 subjects. And then this whole data was so we was anonymized, and then we had a dedicated database at the University of Buffalo where the data was uploaded and then we could process it. So it used RedCap software, yeah, so a RedCap platform. Okay, so now in terms of the how the clinically the consciousness was classified, so this was estimated by a board certified neurologist, and they followed this 4 score, which is the full outline of unresponsiveness. And what this does is this classifies the person in kind of different levels of consciousness. So one level is coma or vegetative unresponsive ecmosis syndrome state. Then there is this minimal conscious state minus, minimally conscious state plus, and emerged from minimally conscious state. So that's like the 4 levels. And we were trying to see if we can kind of classify them based on fNIRS EEG or brain imaging. Again, so this is how the clinically the patients were evaluated, and they used this visual and motor force subscale. So this 4 subscale has also total score that wasn't used. Only the visual and motor as part of that score was used for this classification. Okay, so now for the brain imaging, we used a low density because we wanted to test the feasibility of a low density system. Of course, a high density system is not feasible. Already there were studies that shown that high density EEG very difficult to perform continuously over a long period of time in a ICU setting. So we only had 8 channel of EEG and 8 channel of fNIRS. And then this was recorded simultaneously, both of them recorded simultaneously in 15 minutes segments for 45 minutes to an hour at bedside. So this was a resting state data collection. And then for the EEG, we had this kind of prefrontal areas, FP1, FP2, then F3, F4, C3, C4, P3, P4. So we kind of covered as much as possible prefrontal, frontal, central, and parietal areas. And then with fNIRS, we had like the prefrontal area covered and also the central area covered because we wanted to kind of look into the sensory motor area and also the prefrontal cortex region with a low density system. Okay, so now, so again, this method is not, so we didn't do an exhaustive analysis of which method works the best. So what we did is identified a method that was used for sleep studies to detect arousal during sleep, and then tried to see if that method provides us an estimate of consciousness. So this is this method called AMICA, which is the adaptive mixture independent component analysis. Now, one good thing about this AMICA is that so EEG usually has a lot of artifacts. So we need to anyway do ICA to remove artifacts from the EEG, and different ICA techniques are there. I'm going to discuss that in the next slide. But AMICA is the best in terms of how it detects or how it estimates the sources. These are like more neurophysiological in terms of its sources. So AMICA, that's why is provides us the sources, and then one can then identify any artifacts, remove the artifacts, and then the AMICA then goes into uh, uh, building multiple models of ICA. So by that, what I mean is that, like I mentioned earlier, so the consciousness kind of fluctuates. So the sources may change with this fluctuation in consciousness. So one ICA model may not be good for the whole data. And this is something that the ICA uh, Amica provides is that it actually learns different models that then are combined together in a kind of a Gaussian mixture to then explain the variance in the data. So in that way, you get multiple models, and then with each model, there is a likelihood of that model, how well that model fits the data. So that likelihood can then be used to kind of see how the models are switching from one to another in different patients. So this was then used for classification. And at that time, Amica was kind of already a little bit established in the sleep domain. So that's why I wanted to test it for the in the consciousness coma domain. Okay, so now here are some more detail. Um, so what this ICAs are doing is like they are trying to identify sources that are independent. So by independent, you can have different definitions. So one is this mutual information definition. So now Amica, as you can see on in the plot on the top, is kind of the best in terms of reduction of mutual information between the sources, the ICA components of the sources. And then the ICA components of sources have a neurophysiological meaning. So they are considered to be kind of a cluster of neurons that are active in the brain, and one can do source localization on those components to identify where they are located in the brain. So now the sources are neurophysiologically to be considered have to be dipolar or near dipolar. So these Amica also does the best in terms of identifying sources that are dipolar. So that's why Amica is good in kind of identifying sources, and this is more important for artifact rejection than for classification. But then we wanted to have this one ICA technique that does good for artifact rejection, and then we can also use it for classification. Now, how we do classification. So now what we considered is that so just like in sleep, so in coma, the person may be fluctuating across multiple stages of consciousness. And this is where we need to kind of divide the time series into these clusters where the patient or the person is. And then as they transition, the likelihood of those clusters will change. So this was the idea, and this is what Amica provides. The Amica decomposes the data into these clusters and then fits an ICA model in those clusters separately. And then those ICA models are then kind of considered uh, how much they fit the data in terms of likelihood. So you can imagine of cluster one coming in at a certain time point, then cluster two coming in at another time point, then cluster three coming in at another time point, and then they kind of, I will show you some results, and this is how this data is modeled in this Amica. And then, of course, once you have those likelihoods, then you can use them to see if you can classify different stages or also between vegetative state or unresponsive wakefulness syndrome state and the conscious state. So that's based on EEG. So Amica is an EEG technique. We also looked into this neurovascular coupling because that was kind of also our hypothesis for recovery as a biomarker to monitor the recovery of consciousness. So we looked into the wavelet coherence and the wavelet cross spectrum. So this is where so this is again also not a new technique. So there was already some literature that provided us evidence that this is something that in acute brain injury may be a biomarker, mainly from neonatal work. So what we did is we looked into this wavelet coherence between the EEG band power in this 1 to 12 hertz region. So and then we looked into this with the functional near-infrared spectroscopy data, but mainly the oxyhemoglobin signal. So this is a signal which is very low frequency, around so it's usually around 0.1 hertz. And so there is this wave called Mayer wave, which is near 0.1 hertz, and then you have this fluctuation in resting state around that frequency. So we picked that 0.07 hertz to 0.13 hertz of the oscillations in the functional near spectroscopy oxygenation oxyhemoglobin changes, and then looked at the wavelet coherence between that and the EEG. And again, we used Morlet wavelet. Again, this wavelet was used because of the prior evidence. So we didn't like really here wanted to do any fundamental studies, but it was more an application to see if this is also applied in our case or not. And then the threshold for neurovascular coupling was also determined using a statistical test that was already published. This is a neuroimage paper by Weigert and colleagues in France, and then we followed that paper. So all details are there in our paper. And then, of course, so the phase of that wavelet cross spectrum provides us also this time lag. So whether the hemodynamics is leading the EEG or EEG is leading the hemodynamics. So that was also a scientific question that I wanted to answer, because neurovascular coupling, the way it's defined is that the neural activity is producing the hemodynamic changes. So this is, but then also the scientific question is whether the hemodynamics is affecting the neurons or not. So that kind of a, we could look into that interaction using the phase of the wavelets cross spectrum. So these were some of the ideas that we tried. Now, now for the results, one interesting the aspect that we saw is that, so in healthy, for example, we all know that the EEG power mainly stays at the back of the head. So it's the posterior distribution of, especially the alpha power. So it is at the back. And then in these patients in ICU, so there was this power distribution mostly in the frontal areas, and also, so these are unresponsive patients. Okay, so these are like unresponsive or low responsive patients. The fourth score less than 7. So the power was mostly in the frontal areas. And as you can see, there is this, the lower frequencies are more on the left side, and then the higher frequencies on the right side of the prefrontal frontal cortex, mainly prefrontal areas. So, so this was an interesting observation, and we actually, in fact, are now looking into that more from the scientific, to kind of look into scientifically to answer some scientific questions. But was one observation. And then the second observation was this kind of change in the states, which was, we already expected this. So the multimodal Amica. So while 5 models explain more than 90% of the variance of the data, that kind of shifted for a long recording. So you can see here, like the blue, red, orange, and then there's 2 other colors. So we have this 5 models that are represented here, and these are the probability of a model being active. So this is the likelihood from the EEG data. So this is only EEG data. And what you see is that initially there's blue and then red, then orange, and there's this kind of an oscillation or shifts going on. Not really oscillation, but a shift going on over a period of time. And these were interesting, and I will show you how we use this to classify the patients. But this was an interesting observation in this feasibility study. Now, when we looked at this, how this models come on and off, we found that model 1 and 2 was primarily active in the conscious controls. So what you are seeing on the right is that, you have this probability of the model being active shown in this colors, this 1 dot. For example, at any time point, what's the probability of the model 1 active? And then what's the probability of model 2 active? So that's X and Y, and then that gives you one dot. So similarly, you can do model 1, model 3, model 2, model 3, and then you put those for the whole time series. So you get this kind of a line that you see here. Now, you see 2 colors. One is the green and one is the red. So what the red is, red is the class false. The false is that the subject here or the data here is shown for conscious control. So it's false for being in the unresponsive or low responsive state. And the green is where the class is true, which is what we want to identify is this responsive, unresponsive or low responsive state. So you can see when you plot model 1 and model 3, there is some activity going on, mostly in the green. So the true class, and then the false is kind of a diagonal, so line. Interesting, when you look at model 1 and model 3, you see a separate cluster. So you see that so the model 3 is mainly in the true, so it goes all the way from 0 to 1. And then the model 1 mostly stays. So model 3 is very small, and model 1 goes all the way from 0 to 0.9 for the class false, right? And then the green is all the way in model 3 goes from 0 to 1, but stays quite low on the model 1 probability. So what we saw is that how these models come on and off during this observation, during this in this time series, that we may be able to use that to detect the level of consciousness. So that was this is again an exploratory study to start with. So now we have a hypothesis-driven study at the next like phase two trial. But then yeah, in phase one it's completely exploratory. So we looked at the data and then tried to identify biomarker. Now another aspect that we was interesting is this neurovascular coupling. So what we found is that if you look into this neurovascular coupling, um, so right after brain injury, when they come the first day of the person in the clinic, in the ICU. So the state of the neurovascular coupling right after injury is very predictive of how well they will recover. So so here, for example, what you're seeing is on x-axis are days 1, 2, 3, and 4. And on y-axis is this wavelet coherence, the mean and standard deviation of the magnitude. So so those two um, patients who are shown with this violet and blue, you can see them. So they kind of started below point 0.2. I don't know if you can see my cursor. So yes, it's below point 0.2, and then they recovered. However, they didn't recover consciousness in 3 days. And then their life support was removed and they expired. Now, those who were in the, you can see on above point 0.2. So this is the first day of our evaluation. So they were above point 0.2 in terms of their wavelet coherence, the magnitude. They actually recovered consciousness, and two of them recovered in the second day, and then the blue person recovered on the fourth day. So you can see that so this is again an exploratory study. So we are now have powered it to show to identify this, identify or not identify this as a biomarker. So we have now a powered study. But at this stage, we kind of found that this may be an interesting feature that should be explored. And this kind of aligned with the clinicians' idea that this may be a biomarker. Okay, so now more robust analysis. So when we now look into the classifier, so we have this. So I showed you that this 5 models gives us some idea about the level of consciousness. But then if we do if we develop an actual classifier, and here we did this weighted k-mean, k-neighbor, k-nearest neighbor classifier. And with this, I think it was a 10 cross-validation. So yeah, and then this was using the Euclidean distance metric. So for the k nearest neighbor classifier. So this what this does it like. So why we need KNN? So here we needed KNN because you have the EEG features and then you have the neurovascular coupling features. So this kind of create a kind of a high dimensional space because you have like this 5 likelihood features and then you have this 2 neurovascular coupling channels. So it's kind of a high dimensional space where we wanted to see if we can identify clusters of different patient groups that are being labeled clinically with this 4 score. So there were, like I mentioned earlier, 4 levels. So 4 labels that clinicians gave us. Now, what we found is that, so you can see on the right, the table here. So the first is 1A. So we used this Canaan classifier to separate unresponsive patients from conscious control. So this is only 2 classes here. So we didn't go for all the 4 classes, only 2 classes, unresponsive and conscious controls. And we only used EEG. So if you only use EEG, you get a true positive of 82%, false positive 15%. Now, if you add NIRS, that is the neurovascular coupling feature to that, you increase the true positive to 93%, and then you decrease the false positive to 12%, a little bit. So this was when you classify unresponsive patients from conscious controls. Now, if you want to then look into this classifier to identify unresponsive patients in ICU versus those who are without, so this is the conscious controls are those from the step down unit, and then you have in ICU all the patients who are unresponsive. Now within the ICU, if you look into the unresponsive patients and how well you can classify unresponsive patients in that group of patients in the ICU using EEG only, then you do actually 84%. And then the false positive is 18%. So you have like more false positive. But if you add NIRS, near infrared spectroscopy, so this is where the neurovascular coupling comes in, you do actually very well. So your true positive is 99%. And then your false positive is 1%. So, and I think why this is the case is because in the ICU, in the acute setting, this neurovascular feature actually is very important. But as the person recovers consciousness, or those who don't have that kind of a severe injury, who are in the step down in it, there the neurovascular feature is not that important because most have actually, I think, a reasonable neurovascular coupling. So this is something that we observed in this data. And then also we tried, our original goal was to classify all the four states, which are this coma state, conscious controls, then MCS, ECS state, so minimally conscious state, or emerged from minimally conscious state, or the vegetative state, or unresponsive ecmonal syndrome. So there are like four states. And then if we do that, then we do reasonably well. So you can see this positive predictive value of 85%. And for coma, for conscious state, 87%, for MCS, ECS, 93%, and then for vegetative state and unresponsive ecmosil, 99%. So it does reasonably well. What it does very well when you add FNIRS with EEG is this recovery of consciousness in the acute setting, first 3 days or 4 days after the injury. So this is what now we have powered for our clinical trial. So yeah, so now I'm going to, I think, almost at the end of my talk. So for the discussion, what we used is we didn't reinvent the wheel. So we used a commercially available low density FNIRS EEG bedside device. What we did is we applied this in a clinical to solve a clinical problem. So that's the, like I started in the beginning, that this is a translational research. So we are essentially, it's a solution that we have to develop with the technology that is already available, mature enough for this application. So we identified this FNIRS EEG and then used that to provide a solution to the clinicians to monitor consciousness in the ICU setting. And then we also, so the machine learning, like I said earlier, can be improved, and we are doing that now because this was just this one. We were more interested in kind of showing the feasibility. So it was not optimized, however, still could do quite well. So like I said, unresponsive patients from conscious control, you could distinguish with a positive predictive value of more than 90%. And then also you could classify the levels of consciousness after brain injury using this 4 score. So these were labels were from 4 score quite well. So with positive predictive values between 85 to 93%. So you could do quite well. Now, one challenge here is that we used 4 score, which is, so I will go in that later in terms of limitation. But the labels here are from 4 scores. So this is also an important aspect of the study. Now another aspect is this: how well we could differentiate unresponsive patients with recovery of consciousness. So this is where the patients are in ICU, and then we could distinguish the unresponsive patients from those means like part of them who recovered consciousness quite well. And this was actually with a positive predictive value of 99%. So this is what actually we are now focusing on, being the strength of this neurovascular coupling method. Okay, so now, yeah, so again, limitations. This was small subject group, convenience sampling. So this was not a randomized control. And then some subjects were monitored for several days because I mean, they were not discharged versus those who were discharged or their life treatment was withdrawn, so we couldn't monitor them. Yeah, so that's one of the aspects. So different patients were evaluated for different lengths of time, and also the consciousness assessment was performed using 4 score. And then the gold standard is the coma recovery scale revisited. So we are now using this CRS-R, but the 4 is reasonable alternative because in ICU there are certain limitations, and however, it's more discriminative than Glasgow coma scale. So it's kind of in between Glasgow coma scale and the CRS, the coma recovery scale, which is the gold standard. And also, so we did find that the device that we used at that time, this is an Artemis device that we used, had some interference with the subjects here. So the SNR was different. So this is an aspect that we have now addressed with the new device that is a Nirex Near Scout. So this is a device that we tested in healthy, and we could see that it does well with hair or without hair. So yeah, so this was one of the challenges with this particular device. And that's why we used only the frontal neurovascular coupling. So the two channels that were used for this classifier are the frontal channels, because the channels on the sensory motor area had this hair issue. So the signal, the SNR was not very good. And yeah, so the another limitation, like I mentioned, is an exploratory study. So it was not powered to show that FNIRS EEG, this neurovascular coupling idea, is better than EEG alone. However, that's what we are doing now in the clinical trial. Okay, so now, so this is where the concept selection goes. So we selected FNIRS EEG over EEG only, and of course, this needs to now go through a clinical trial to show that this is indeed the case. Now, I also work in kind of translation where I work with companies, and also one of the aspects that we are now looking into is this translation with a company. So we have filed a patent on this, and then it was started as a study in adults. However, we are now also doing in neonates, and also we want to make this device affordable. All this is a separate topic, but anytime we work with a biomedical device, we need to make sure that that's accessible. So it's not that you have like a device that costs like $500 and you make it like 5,000 or $50,000. So you have to. So this is an aspect that also we are, because these devices can be made quite inexpensively and can be made quite affordable for the global health equity. That's one of the areas that we are working on, and this is the funding that we got from University of Buffalo for this device development. And then currently it's not CE approved, so we cannot use it in Europe or in US. However, I think this CE approval is a way to make things more expensive so that developing countries cannot use. So that's why it's not CE approved. However, we have approval in India to use it in neonates. So it's again a very simple device. Like I said, it has this LEDs, which are of course finely controlled to control the light intensity. And then we have photodiodes, which are also quite inexpensive nowadays, that you combine with light source to do the FNIRS. And for EEG, there are like so many SoCs available, system on chips available that you can use. So this is the device. This is again for babies. It's like a headband that we use, and this is our ongoing trial. Not ongoing, actually, now it has finished. We are now going for the next level of funding, but this was from the Gates Foundation. We got this funding for neonatal brain injury. So this is kind of the wire. It goes to the next step for the commercialization and for the social impact. Okay, thank you very much. So that's the end of my talk. Hopefully I'm within the time.
Thank you, Anir. Let me stop recording first of all.